episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Okay, welcome everybody again to another episode of our show with another truly fascinating guest that is focused on creating a better tomorrow for all of us. On today's episode, we are going to again segue into sort of the fascinating domain of biomimicry, sort of broadly defined as the design or production of materials and structures and various systems that are modeled on biologic entities and processes. As those that watch the show know, a few episodes ago, we spent some time talking about spider silk and meeting with the real-life Spider-Man who's creating super strong fabrics for us. Today, we are going to get into another really cool topic, sci-fi-ish, but really cool nonetheless. We're going to talk about invisibility. We have the honor today of being joined by Dr. Alon Gorodetsky, who is Associate Professor in the Department of Chemical and Biomolecular Engineering at University of California, Irvine, and he has joint appointments in the Department of Chemistry and the Department of Material Science Engineering. Dr. Gorodetsky has his Bachelor of Science degree in Engineering, Physics, and Material Sciences from Cornell, his Ph.D. in Chemistry from Caltech, and he subsequently completed his postdoc work at the National Science Foundation American Competitiveness and Chemistry Fellow at Columbia University. His current research is focused on the development of macromolecular and biomolecular materials inspired by natural systems with an emphasis on cephalopods, namely our friends, the squids, octopus, and cuttlefish. His work has been featured in a variety of popular media, Popular Science, Popular Mechanics, Newsweek, Wired, Verge, Fox News, list goes on and on. Dr. Gorodetsky has received several awards, including the Henry Samueli School of Engineering Mid-Career Faculty Excellent Research Award. He's received the Air Force Office of Scientific Research Young Investigator Award, the DARPA Young Faculty Award with Director's Option, Presidential Early Career Award for Scientists and Engineers. A lot going on here. Let's get right to it. Dr. Gorodetsky, thank you for taking the time out of your schedule to come on the show today and talk to us about these fascinating topics.
Thank you for having me. It's really my pleasure.
Typically, we start the show off. We just hand the mic over to our guests for a little bit, just to talk a little about themselves. If you could sort of take us back a bit, where you grew up, how you got interested in science and chemistry, and this fascinating domain of biomolecular engineering, biomimicry. Would be, I think, a great place to start to sort of lay the groundwork for everything we're going to be talking about.
Of course, yeah, I'd be happy to tell you about that. So I'm actually originally, I'm an immigrant. We left at that time. It was the Soviet Union at the end of the 80s. And I spent most of my let's call them formative years, I guess, middle school, junior high, and high school on the East Coast in Massachusetts, which I'm still very, very fond of. Of course, the sports teams ties, run deep there. So and both my parents were engineers, so they kind of naturally fostered an interest in science and engineering. Although they did switch career tracks a little bit later. But so after kind of growing up in Massachusetts, I went to school at upstate New York at Cornell. And, there I had a really wonderful mentor as an undergraduate student, a scientist named George Maliaros, who's now at University of Cambridge. And he really kind of helped cement and instill a love of science in me as an undergraduate, and a love of research in particular, even though I had that already. And I was working on very different things at the time. At the time, his laboratory and myself in particular, we were focused on photonics, optics, let's say light emitting diodes. And so that was a really amazing undergraduate experience. And then I was doing a lot of engineering, but I kind of had an urge to or an interest in learning more about the fundamentals, doing more basic science from there. Because I was always asking, well, how does this work? Why does this work? Those types of questions. And so from there, when I was looking at PhD programs, I was fortunate enough to be admitted to Caltech, and I visited, and just one of those things that's serendipity, it's chance. I met this another amazing scientist, Jacqueline Barton, who's at Caltech, and she was the head of the division of chemistry and chemical engineering there, or later she was. But she told me this fascinating things that she was doing with charge transfer in DNA, which just really captured my attention. And so that was the way that I transitioned from working more on engineering and physics and materials to more doing biochemistry. And I did work on protein DNA interactions essentially for the duration of my PhD, and specifically the electrical or electrochemical properties of DNA, which was really kind of, I think Jackie's work in that area was just groundbreaking. And so from there, again, there I learned a lot about the fundamentals and basic chemistry. And then, we started collaboration with another terrific scientist, Colin Nuckolls at Columbia. And so I ended up doing a postdoc in his group where I wanted to add some synthetic tools to my repertoire. So the ability to engineer systems, understand them, and to actually synthesize or tweak them at the molecular level. So with that wonderful skill set, right, that I developed, I again, serendipity, a lot of luck. I managed to get this great position at the University of California, Irvine at the end of my postdoc. And at that point, it was a joint chemical engineering material science department, which was very much in line with my part of my training, where I was right at the interface between those disciplines and also doing a lot of chemistry as well. And so the first day I was starting my independent position, so I remember this, because it was kind of changed the trajectory of my career. So, I was giving a talk on photovoltaics, solar cells. That's what I'd worked on in Colin's group. So again, I'd swung back a little bit towards the optics area. And I walk into a seminar being given by a scientist named Roger Hanlon from the Marine Biological Laboratory at Woods Hole. This goes back to your comments about MBL that you made right before we started. And I'm just watching these videos, of cuttlefish and squid and octopus and, these cephalopods just changing color and, matching their patterns. And this very famous video of an octopus that looks like algae on a rock just popping out, right? It's like watching a rock transform into an octopus in real time, right? It'd be like if I transformed, I like to use the example, if I transformed into a file cabinet while we're talking, right? And so right there, I'd done all this groundwork and it's had this vision of what I was going to do for research. I said, okay, I'm going to drop half my research and just focus on materials inspired by these animals. Because again, just blind luck, walking into this seminar, and it was just so fascinating that I had to kind of follow my intellectual curiosity and learn more about it. And so that ultimately, changed the trajectory, I think, of my entire scientific career, because half the work that we had been doing was on, organic electronic materials and, making things that could be used for nanoscale circuits. And then the other half was on this cephalopod inspired theme. And at first it was some of the things we were trying were so different and so unique that it was hard to get it off the ground. But once we established that, it just started growing and growing and growing. And then a few years later, after I got tenure, it basically started to encompass my entire laboratory. So moving forward, that's all we're going to be focusing on, and becoming more and more biological as we go along the way of not just developing materials, but trying to tweak biological systems to emulate or recapitulate some of these key capabilities of cephalopods. Someone once described it to me as kind of the ultimate complementary, or the ultimate complement to nature in terms of reverse engineering. And that's the way that I like to think of it, of taking what is the key concept for how a system functions and then translating that concept to a device or to a cell or to, let's say, a different macromolecule, and really making a material or a system that works in a similar way to nature. So I like to think of it as bioinspiration, because cephalopods are so amazing and so complex that truly mimicking them, I think, is really far away from where we are right now. But we can capture little slices of what they do, I think. So that's the goal. Yeah. So that might give you kind of an idea of the trajectory, at least up until this point. So.
Yeah, I appreciate that. And I like that term, bioinspiration. And the other thing I was thinking about, sort of, you sort of run the sort of that Walt Disney moment that, like, maybe 30 years from now, you're going to tell everybody it all started with an octopus, as opposed to. Anyway, but getting back to that, you mentioned, obviously, cephalopods have been sort of in the public eye quite a bit in the last couple of years. And the octopus has gotten a lot of attention, not just for sort of camouflage, but its nervous system and having sort of multiple brains and all sort of the weird memory and consciousness potential things that we can be inspired by there. Obviously, the squid and the cuttlefish with these chromatophores that once again have these fascinating camouflage abilities. Did a little reading offline. They have various hormones and neurotransmitters, and sort of this ability can be stimulated by how you change its mood or the temperature of the water and so forth. Take us, if you would, just on a little walk through sort of, and so not a long lecture about squid and camouflage, but what are some of the basic things that we are learning nowadays about what's going on with regard to that, turning that process on and off as the squid's hanging out there in its tank or in the ocean? What are some of the key bio-inspirational learnings that help guide your work?
So I think there's been a few things recently in particular. So I think there's been a lot of understanding developed for how the cells in cephalopod skin function, and specifically these cells called iridophores, and a lot of that really interesting and kind of exciting studies in that area were done by the group of Dan Morse at UC Santa Barbara. And then also chromatophores, where a lot of work on that front at the cellular level has been done again by Roger Hanlon and collaborators, of course. So those two cell types are the paradigms typically for the types of things that we try to engineer. There's another cell type called leucophores they're also reflective. They work a little differently than iridophores. And again, both Roger and Dan have contributed significantly to those. So they really do, their research has been the fundamental biology that we have looked to and that we have drawn on consistently for the design of our systems. And what they're teasing out is how these cells function and how they may work together to enable some of these camouflage abilities in really unexpected ways. So one really interesting example a few years back from Dan's group was finding that certain types of female squid have actually tunable leucophores in a stripe on their body, I will say. And that stripe they can turn on and off to mask themselves, from unwanted suitors. And so those tunable leucophores really have some very interesting characteristics that we've looked to for developing mammalian cells that may capture just a little slice of that. And with respect to chromatophores, for example, which Roger has worked on extensively, so about a year and a half ago, I believe, they published this really exciting paper where they show the chromatophores can not only change their aerial coverage, because they expand from tiny little points to large plates to change essentially the transparency of the skin, but they can also change their iridescence. And so this was, again, a wonderful multidisciplinary team. And that has those types of studies, which Roger has been publishing for many years, for these mechanically actuated systems. Chromatophores are actually organs, small organs. They have different cell types in them. And so those have inspired more artificial materials in our lab, which we mechanically actuate, we stretch in order to change how they transmit or reflect or absorb light. So those fundamental studies at the cellular level have been really, really critical. And where we've gone is we've tried to understand the proteins that enable the functionality of both of these cell types, the cell types and the organ type. These proteins, reflectins, at a structural level, and also understand their self-assembly, and specifically understand the relationships between their structures and their properties, their refractive indices, which are unusually high. And then we've also, made infrared reflecting or infrared transmitting materials that are actuated in similar fashion to chromatophores or layers of skin that contain chromatophores. So those studies have been wonderful to draw on. Of course, we don't house squid in our lab, although it would be wonderful to have a tank in the future. It's been a goal of mine for several years.
Gotta have a good mascot in the lab.
Yeah. So we just haven't gotten there yet.
Yeah. But that's a great background. Now, drawing on everything that you're taking from that body of work, you've been very busy. Well, you've been extremely busy the last few years, but this last year specifically, you published in Nature Communications cephalopod-inspired optical engineering of human cells, where you created squid-like transparency in human cells in the lab. And then most recently in the Proceedings of the National Academy of Science, structure, self-assembly, and properties of truncated reflectant variant. Talk a little bit about this research, if you would, and sort of what you've been up to this last year. And then obviously I'm not ready to turn invisible yet, but some of the shorter term applications of having these invisible like human cells in the petri dish, and some of the things that we can use them for in the short term in terms of better understanding of sort of cellular biology or drug discovery, things of this nature. Take us a little on that path, if you would.
Of course. So I will say, of course, one of the things that I didn't mention when I was a kid, I was inspired a lot by science fiction, right? I read a lot of science fiction, comic books, these days, they have all these cinematic universes that have these just wonderful kind of superheroes, everything you can imagine, so some of that stuff takes you back. That it's kind of like being a kid again. So with that having been said, I think I agree with you. We are very far away from adaptive invisibility in people. I even probably, realistically speaking, probably outside my academic lifetime. But I think we can do some really interesting things that have some biomedical and technological potential. Cool. And so typically when people think of cells, right, they're as if you have a cell culture, for example, they're pretty transparent to begin with. So it's hard to look at them through a conventional, let's say, optical microscope and pick up all the details. But what would really be advantageous and what motivated our work was incorporating this tunability. So it's a matter of changing the transparency of the cells controllably and reversibly with respect to the background to make them easier to interrogate, I'll say. And there's a tremendous body of work on that, right? And it's typically for deceased, I will say, cells or tissues. So that's been the primary focus. But being able to do that with a living cell or a living tissue is very challenging, because you would typically fix them, and then you would remove the things that would absorb light, which are dyes, and then you would try to in some way change the refractive index, so you would reduce scattering of light. And then you would get something that is, you know, for all intents and purposes, will be transparent. And a lot of the concepts at play here are similar to this classic experiment where you can take a glass rod and put it in a liquid that has an identical refractive index, right? You know, many people have done that experiment at some point in their educations, right? And you almost can't see it, or some variant of that. So that was the general concept that motivated us to start engineering human cells. And this is all commercially available stuff. These are very standard, you know, standard cell lines that have been the workhorse of this type of research for many, many years. And so we started doing was actually introducing reflectins into these cells using genetic engineering techniques. So you temporarily get them to express almost the same proteins that are found in squid skin cells, except you have them expressed or produced, I'll say, in a human cell. And what was really the aha moment there was in squid skin cells, they form very particular and very specific type of structures that have these light scattering or light reflecting properties. And when we introduce these proteins in large quantities into human cells, the structures that they formed superficially, at least morphologically, were almost identical to those that you'd see in squid skin cells, which really was just remarkable. I mean, that right away, as soon as we saw these microscopy images, we knew we really had something, and I was very confident. You know, even before doing all the ancillary experiments that you need to prove this, it was just so shockingly similar to what you see in squid skin cells that it was really exciting. And then we started probing the cells, and some of the properties in terms of scattering of light that you would have in the squid skin cells, specifically the tunable leucophores that are found in cuttlefish, or not in cuttlefish, excuse me, the tunable leucophores that are found in female squid, and then the more static leucophores that are found in cuttlefish. And so they were recapitulated, at least to some extent, within these cells, the human cells, and we could tune the degree of transparency using an external, fairly gentle chemical stimulus. So essentially, you just change the salt concentration of the surrounding solution. When you grow cells, they're in a very, you know, a mixture that has a very, very high ionic strength and a lot of different things in it, a lot of salt. But if you just increase one of the salts selectively, you could change the scattering of light from these cells. And we could demonstrate that at the single cell level, and we could also demonstrate that at the cell culture level. And so with that, you essentially have cells where you can change the way the protein aggregates in their interior, change the way they scatter light, and then because of that, change their transparency. So light scattering due to differences in refractive index is one of the things that dictates transparency, and doing it in a living system. So that has a lot of potential for visualizing different biological processes. So, you know, just like analogously to how fluorophores and fluorescence microscopes are one of the key technologies that enables you to study what's going on inside a cell, inside living systems. You know, you stain them, and then you try to follow one of these tags along. So you could imagine using these high refractive index proteins as similar tags to look at biological processes, but they might have the advantage that, you know, they're not going to bleach the way that a fluorescent probe will, which is lose their ability to fluoresce over time, and you know they could allow you to potentially see certain cellular structures better or more effectively. You know, of course, you have to prove a lot of that stuff, but I think we have the groundwork for doing it. and so of course, you know, if you want to do any of this, right, and those are just the nearest term, you know, biotechnological applications that we're thinking of. You really have to understand how the structure of this protein, or you know, weak structure, I would say. So it's partially ordered. This partial order of this protein would determine these properties that you then want to harness for an application. And that is the latter paper that you mentioned from in the Proceedings of the National Academy of Sciences, USA. And there we took a little segment. So we kept going smaller, right? We took reflectins, you know, from the cells, we took a reflectin, and then we took a little segment of one of the reflectins. The same one that we use for cellular engineering, and actually tagged it with different isotopes, and learned how to control its self-assembly in solution. And that gave us the ability to solve its structure, or in certain regions, understand that it had a very dynamic or a lack of a structure, using a variety of techniques, you know, computational methods. And this was a terrific collaborative team. You know, I really give all credit to my colleagues and coworkers. And so through that combination of computational techniques, X-ray scattering methods, nuclear magnetic resonance spectroscopy, I don't want to try to avoid getting too technical. What we got is essentially what the conformation of this segment of this protein is in solution. And then we used another very straightforward method for controlling its self-assembly and structure. And so this was a discovery by one of my students, where she just shook the protein. You know, you take a little vial and you basically shake it for say 6 hours, right? You typically wouldn't do that, but in this case we did, right? And then all of a sudden you'd see it self-assemble into things that look like you know, beads on a string, or kind of like, you know, let's say beads on a noodle, something like that, and then ultimately into little fibrils. And those fibrils seem to be very similar to beta amyloid type fibrils that actually cause, or are one of the mechanisms that are associated with that self-assembly is associated with Alzheimer's and other neurodegenerative disorders. So again, that was very surprising, the protein. But what was interesting is, as you change the structure of the protein, as it became more and more structured, you also change its refractive index.
Okay.
So, and we measured that directly, not only the refractive index, but how these nanoparticles, and then beads on a noodle, and then these fibrils, how they all scattered light. And so essentially by shaking this protein, you can change how it scatters light, and you can change its refractive index, which is related to the way that light propagates through the material. And so that was really fascinating. So going back to what I mentioned earlier about chromatophores going from tiny little points to large plates being mechanically actuated, right? So now you have a mechanical mechanism for changing the structure of a segment of this protein, and potentially the full length protein, and then changing its refractive index, right? So maybe, you know, and this is a far out there idea, maybe you can take a vat of these cells, shake them really hard, right? Get the protein to change its structure and change its transparency. You know, we haven't quite done that yet. I don't know if it would work, but you know, I like these kind of crazy concepts. Yeah. So these are two foundational pieces for the future biological work in my lab, right? So you go really small, you understand the structure of the proteins, you understand the structures that you have inside a cell, and then you can try to build back up, you know, what happens to a cluster of these cells, what happens to a tissue, right? And then see what type of control over optical properties you can get. So that's the direction that we're heading in. And then if you have a tissue that can change transparency, well, there's a lot of really good fundamental science you can do for living tissues, I think. You know, and again, you know, these are very lofty goals, right? Hey. You know, if you've seen those pictures of the transparent mice and the transparent brain, right? That's like so far beyond for for these static disease systems, so far beyond what we've done right now. But it's, if you can get, let's say like transparent mouse skin or something along those lines, that will be pretty neat, right? And there's precedent in nature for that, right?
Sure.
If you've ever seen these frogs that their skin is completely transparent, you can see their internal organs.
Yep.
Right. So I don't know if you capture that in another biological system. I mean, it's a great question to think about, right? And it's, if nature can do it, maybe it's not so far out there.
No,
right?
I mean, look, it's evolution has been perfected this stuff, so there's a reason it didn't disappear, and then
right.
It's just a matter of tinkering a little more, and
right.
It'd be really fascinating. See what you come up with.
That's why I have this green wall behind me as an example of foliage, right?
Right.
If you had a frog with transparent skin, or that could match in there, that could match to it, it'll be very, very hard to pick up.
Yep.
So yeah, so it's just conceptually, it's such a fascinating area. Yeah.
As you're talking about, obviously you're moving up this chain, you work on the proteins and the variants, you work at the cell level. I read a little bit about, I think it was last year, because you've been involved in a lot of things. It was an article actually, and it was talking about the heat regulation properties of these chromatophores, I think it was, as well. And I know sort of temperature is somewhat related to the invisibility component, but the article specifically was talking about some sort of next generation fibers or fabrics that potentially could be inspired by some of these techniques. What was that about?
Sure. Yeah. So that's the other branch of my lab. That's more as I mentioned earlier, I have this need to understand things, but as an engineer, you also
Sure.
want to make something practical.
Sure.
So that's where I think really the bioinspiration part really comes into play, because a cephalopod underwater, do they really need to manage infrared radiation with their skin? Probably not so much, right? But if you can take that concept and translate the same structural characteristics and the same structures in the way they work to a material that works in the infrared, then you really have something. And so what we did is we thought of how one of these layers of skin that has chromatophore cells embedded in it really works. And the way to think about it, and this was figured out, of course, not by ourselves, but the way to think about it is you have this transparent sheet, and you've got these colored little points or spheres embedded throughout it.
Mm-hmm.
And then when these spheres are ringed by radial muscle fibers, so these muscle fibers will pull them apart into a plate. And then you can imagine the aerial coverage, if they're expanded in area by 10, 20-fold, right? From a tiny point to a plate.
Yep.
And so if you do that, you completely change the transparency of this layer of skin, this sheet. And so that concept, plus this, the classic space blanket technology, or the Mylar blanket, which everyone knows about.
Sure.
It's just a piece of plastic with a coating of metal on it, right? I mean, the inside of a potato chip bag is essentially the same thing, right? So we took those two ideas and developed a material that was a hybrid of them. And the hybrid was having a stretchy, very thin, transparent piece of rubber, and then putting a metal layer on top of it. But then having that metal layer be able to be reversibly broken apart into islands. And so then you spread these islands apart, and in the spaces in between them, you only have the transparent rubber. So then infrared radiation can really go through. And that's analogous to having these small points in that layer of squid skin. And then when you let these islands come back together, you release the rubber, so they all come back together. then you get this solid infrared reflective kind of shiny film that's very analogous to the static version, the space blanket, which, you know, these aluminum films are almost perfectly reflecting. And it's analogous to the expanded chromatophores, which are plates, which cover, you know, would have complete aerial coverage or almost complete aerial coverage over that formally transparent layer of squid skin. And so you have something that is tunable, like that layer of skin, but that has the technological properties and capabilities of the space blanket in terms of modulating infrared radiation. So you can control the degree of infrared light or infrared radiation or heat, you know, however you want to frame it, that's both transmitted through and reflected from this space blanket and squid inspired material. Right. So, and we really proved this very systematically. You know, you can take this material, you can take the space blanket, you can put them under identical conditions. You know, you can test them the same way, benchmark everything against the space blanket, and, you know, a metal layer on a polymer or a metal layer on rubber pretty much work the same way. But then when you can break it up into these islands and spread them apart from each other and then bring them back together, then you have a tunable variant of the space blanket.
Got it.
Right. And so where that could come into play, right, is, you know, we're both sitting here, you know, I'm in California, you know, you're in Philadelphia, if I'm not mistaken, right?
Mm-hmm.
And
Correct.
the weather there is very different.
Sure.
Right. You know, and so from here, that is, right. And so if we wanted to wear the same type of shirt or type of clothing, the ability to tune it to trap the infrared radiation, keep you warm in your case, and to release the infrared radiation, say from my body, and keep me cool, in my case, that will be a very powerful technology. And so that's what our initial work has effectively demonstrated. And so with that in hand, you know, there's a lot of development that has to be done. And one of the challenges that we're really working on now is, can you scale this stuff? Mm-hmm. You know, people spend all this time developing these amazing things in the laboratory, right? But you know, the devil's in the details, and you go, okay, I want to have a tarp made out of this stuff, right? Or I want to make it in gigantic rolls or in a batch or something, right? And how do you get there? And so that is one of the things that we're also focusing on, you know, scaling our materials so to enable a lot of practical applications. And we've had some success with that recently. So I think that will open up, you know, not necessarily more fundamental studies, because in terms of performance, I think we really maxed out the material. But in terms of the types of applications that you can go after, you know, and I probably don't need to belabor them, right? If you know, if you've ever seen, let's say, a marathon runner cover themselves with a space blanket afterwards, or anything that's shipped in, you know, this metallized, you know, in a metallized plastic foil, basically, yep. Right? You know, those applications are already there. So I think we can add that tunability aspect to it, and that's what's really exciting. So your questions are hitting at kind of my two favorite things, right? Really understanding something in extreme depth, and going all the way down to the molecular level, so you can truly engineer the system across multiple length scales, but then also, you know, trying to do something that will be a technology and that will be relevant. So I guess that's my kind of hybrid, you know, split background in both fundamental science and engineering, right? It's hard to run away from either one.
Yeah, but it's you know, these things go hand in hand, and they're extremely important. And you know, you're bringing up an important point about, you know, sort of scalability, and you know, we've done some episodes in the past where we talked about sort of not artificial, but sort of lab engineered meats and so forth. But yeah, you can make a $250,000 hamburger today, But obviously it's going to come down, and the same thing with this. I mean, obviously you've made tremendous strides, and the fact that you have sort of both parts of this, that you can go into the sort of the reductionist detail of the proteins, but at the same time think about systems on how this comes together as an engineer, and I think it's great. I mean, it's very exciting, and I just, I see the. I'm more so the pharmaceutical guy, but I see the potential for where this potentially all goes.
I think one of the things that's really important that you learn as you go along is to really focus in understanding an area in depth.
Yep.
And not spreading yourself too thin so that you do only superficial studies of systems. So I think that's why we focus so much on in the other areas on cephalopods that have become more dominant, on really nailing the details and so to speak, and really just hammering away at that basic understanding, because you can never develop great technologies if you don't understand what's going on, right? And that was another reason that drove us to just refocus. And of course, the students strive. They have to steer the ship, right? So in some ways, the interests of the students is what determines where you go.
Yep.
So that too has played a pretty significant role.
It's a fascinating story. Wishing you the best with it moving forward, because it's just, as mentioned when we talking offline, I love science fiction as you do, and to see it materialize like this and to bridge it from science fiction to science fact is, but what about all the shows about sort of creating the future and a better tomorrow? So it's been a really great time for everybody that's going to be watching this episode on the YouTube or listening on the podcast. You've been listening to the amazing Dr. Alon Gorodetsky, Associate Professor in the Department of Chemical and Biomolecular Engineering at University of California, Irvine, doing truly amazing things in the development of these macromolecular and biomolecular systems inspired by our friends, the cephalopods. Alon, thank you for taking the time out of your schedule to come talk to us for a little while. Thank you for everything that you're doing and continue to do, and for your future research plans to move this forward. And as we say, thank you for creating a better tomorrow for all of us. Really see the amazing potential here, and it's just really wonderful. So thanks for all that.
Thank you so much. It's been a pleasure. Thank you for having me, and thank you for your time as well. It's great. The series you put on is really just fascinating, and can really help people learn about fields they might have never thought about before. So.
Absolutely, that's what we try to do. Great seeing you.